So let's first start out by talking about plastids. What are they? Plastids are organelles that are unique to plant cells that vary in both form and function. They all start out as undifferentiated proplastids in the meristem, but then they specialize into all these different cell types, or plastid types. The most common plastid type, which I'm sure you all have heard of, is the chloroplast. Now, as we all know, cells differentiate, and we can examine this using a variety of different methods. We can see this histologically based on the morphological differences between different cell types, but we can also infer this based on the transcriptomic state of each cell. In other words, by studying the RNA content and gene expression levels within each cell, we can actually deduce the cell type. And we can do this using single-cell RNA sequencing technology. Now, as cells differentiate, plastids also differentiate. But very little is known on these plastid differentiation dynamics, especially in the roots, because it's very hard to study these dynamics because plastids in the roots have variable morphology and there's a lack of pigmentation. But using single-cell RNA sequencing technology, we can actually study these plastid differentiation dynamics, specifically their timing relative to cell differentiation. Now, along the root of Arabidopsis thaliana, which is the plant type we'll be using in this study, we see a variety of different cell types as indicated by the colors on this diagram. Also, we get a gradient of differentiation along the length of the root, where older, more mature cell types are found nearer to the top of the root, and younger cell types are found in the meristem, which is this region of undifferentiated cells found right above the root cap at the tip of the roots. And using single-cell RNA sequencing technology, we can capture gene expression profiles for each and every one of these cells. And we can study their development, specifically plastid differentiation relative to that development. And we can begin to answer one of the main questions of this study of whether plastids are heterogeneous within cell types. In other words, do the same cells have the same plastids? Now, very little is known about this in the literature. Basically, what we know is we know that proplastids are characteristic of meristematic cells, and amyloplasts are found in the columella cap cells. But very little else is known on the types of plastids in all these other cell types. So that's something we'll try to study in this experiment. Do the same cell types have the same plastid types? Another thing we want to study is plastid state coupled with cell state? So as we look at cell development and plastid development, do we see this linkage and synchronicity between the two processes as indicated by this dark blue line here? Or do we see no linkage at all, where it would be a sudden increase in plastid development somewhere along that cell differentiation pathway? Another thing to note here is that we'll be using meristematic cells and columella cap cells, as those are the two types of cells that have characteristic plastid types. So meristematic cells have a higher expression of proplastid genes, and columella cap cells have a higher expression of amyloplast genes. So again, the two objectives of this study: do the same cell types have the same plastid type, and is plastid state coupled to cell state? Now let's get into our methods and results, which will involve a lot of clustering and pseudo-time analysis, but we'll get into that soon. So the nature of the data in this study, basically it was published single-cell RNA sequencing data of over 7,500 plant root cells, again from Arabidopsis thaliana. And the first thing we wanted to do with this data is we wanted to cluster them. So we used a dimensionality reduction technique called UMAP. And basically, about this plot, each dot here represents a cell. And it's thought that cells are more related to cells are more related to the cells within their clusters than cells outside of their cluster. So as we can see, we see all the different cell types from that earlier plant root diagram, and we even see some additional cell types with the phloem and the protoxylem, which are actually subpopulations of the stellae. Now, we did the same thing with plastid types to see if we can cluster those different plastid types. And we clustered based on 974 nucleus-encoded plastid genes from the plant proteome database. So by clustering based only on those genes, we wanted to identify these different plastid types, or plastid classes. And we were able to identify 11 different plastid classes. And now using these two clusterings, we wanted to see if we could answer one of the main questions of the study of plastid heterogeneity by trying to match up plastid type clustering to cell type clustering. So we got plots like these, where basically we highlighted individual cell types on each of these plots. I'm showing 3 plots here. But if we take a look at this first plot on the left here, basically we highlighted the stellae cells, and we can see this exclusivity of stellae cells within one of the plastid clusters. So this suggests that plastid types, that most cells of the same type have the same plastid type. But we still do see some red dots scattered over the plot, so this does suggest some plastid heterogeneity as well. And by identifying the cells, the cluster that contains meristematic cells and columellar cap cells, we were actually able to identify those cells as proplastid-containing cells and amyloplast-containing cells, as we mentioned earlier. And why is this exciting? Well, we can actually, by finding this cluster, we can find differentially expressed genes for these proplastid-containing cells and amyloplast-containing cells, and we can develop a list of putative marker genes for these two types of plastids, which is a gene expression, which is a way to use gene expression to identify these cells, which is absent from the literature. And actually, by identifying these, we also are able to define plastid state, because if a cell has a high expression of proplastid marker genes, then it's probably earlier along that plastid differentiation pathway. But if it has a higher expression of amyloplast genes, then it's probably towards the end of the pathway, because these are more mature plastid types. So now that we have plastid state defined, how can we define cell state? We can use a method called pseudo-time analysis, where basically cells are ordered based on their gene expression profiles. So basically, we did this pseudo-time analysis based only on non-plastid genes, so as to define cell state independent of plastid state. And what we get is we get these undifferentiated cells, which are the meristematic cells. We get them at one end of the trajectory, and at the other end, we get the columellar cap cells. So now, why does this define cell state? Well, it's because this dyno package, which was used to do this pseudo-time analysis, actually gives a progression percentage for each cell along this trajectory, where lower progression percentages are indicative of cells being earlier along that trajectory, and higher percentages are indicative of cells being towards the end of the trajectory. So now that we have cell state and plastid state defined, what happens if we plot these against each other? What do we see? Well, at least for proplastid gene expression, what we see is that we see a significant decrease in proplastid gene expressions as cells differentiate from meristematic cells to columellar cap cells. And for amyloplast gene expression, we get a significant increase. So this is good. So as cells become more mature, their plastids also seem to be getting more mature. But this gap in the middle here makes it a little difficult to infer synchronicity, which was one of the objectives. So how did we look deeper into that? Well, we divided the pseudo-time into 3 stages. First stage was from 0 to 20%. Second stage was 20 to 70%. And third stage was 70 to 100%. And well, for proplastid gene expression, we got a significant decrease in proplastid gene expression in all 3 stages, which probably indicates synchronicity between proplastid gene expression and cell state. Now, for amyloplast gene expression, we get a different story. Amyloplast gene expression only significantly increased in stage 2, which is probably an indication of less synchronicity. Now, to wrap up, this study gives insight into the question of plastid heterogeneity. We were able to find that most cells of the same type have the same plastids, but we still do see some indication of heterogeneity. Also, our results suggest that at least for proplastid gene expression, it is in fact coupled with cell state. But further research will be necessary to actually confirm this. For example, cells at the end of the trajectory need to be verified as actually more differentiated than cells at the beginning of that pseudo-time trajectory. I'd like to acknowledge the following people: my wonderful mentor, Tessa Green, my tutor, Deborah van Egeren, and also the entire Deborah Marks' lab at Harvard Medical School. I'd also like to recognize my sponsors and thank MIT, CE, and RSI for giving me this wonderful opportunity.
We will now take questions from the judges.
So I'm not familiar with the UMAP algorithm, but some of the other algorithms that are used for dimensionality reduction in the omics world, like t-SNE and whatnot, have parameters that you can set, and if you set them wrong, like the neighborhood size, for example, you can get false clusters. So I'm just wondering what you did originally to convince yourself that the clusters you were seeing in your reduced dimensional space actually mapped onto real biological entities.
Yep, so the question is asking about the UMAP algorithm. How are we certain that these actually match to actual cell types? And actually I have a supplemental slide on that. Basically, what happened is we used a resolution of 0.8 to cluster these to cluster the cell types, and we got originally 20 different clusters, but we looked at marker gene expression data for each of the cells. So for example, this gene is known in the literature to be a marker gene for endodermal cells, and we found that cluster 9 of the original cluster matched up, as you can see by this high gene expression here. And then we found some cell types, like the lateral root cap, that aligned to 2 different clusters. So these were assigned as both as lateral root cap clusters. So we actually identified these clusters based on that, and we grouped them if we found similar gene expression within clusters.
So I'm curious, when you do your pseudo-time trajectories, it looked like you picked out some of the clusters somehow. What was the methodology from going from this bigger dimensionality reduction to these smaller clusters where you were doing that path?
So the question was about how we chose the meristematic cells and the columellar cap cells to do that trajectory analysis. And how we chose that was those are the 2 cell types that are known to have 2 characteristic plastid types. And so we don't want to choose like endodermal cells, which it's not really known what type of plastids are in those cells. So by choosing those 2 cells and looking at the development of cell state and plastid state, we were able to try to answer the question of coupling. So.
And just to be clear, the data you start with is just gene expression and the cell types, nothing about plastids, or is there some additional data as well?
Yeah, so for the pseudo-time analysis, we took out all the plastid genes because we wanted to find cell state independent of plastid state.
Can I go back to one of your early slides where you were laying out mutter?
Mm-hmm.
That, right?
This?
This one.
Mm-hmm.
So when you have on your x-axis, your breast and cornella, that is pseudo-time, or what are you actually using for your variable there?
Yep, so the question is about the variable used on the x-axis from meristematic cells to columellar or cap cells. Yeah, that is the pseudo-time. So basically those are the progression percentages for each cell given to us by the dyno package, which ordered the cells from undifferentiated to differentiated, or at least assume so.
And what you actually saw was nothing in the middle.
Yeah.
Effectively, it's cell clustering on either side.
Yep. Yeah, so that was kind of why we did the 3 stages thing. Because of that gap in the middle, we weren't able to infer synchronicity because, I mean, true synchronicity would be throughout the entire process as cells are developing, plastids are developing closely linked to that process. So with that gap, it was a little hard to infer synchronicity. And by dividing it into 3 stages, we were able to see, is it just because of a sudden jump, as would be seen by this light blue line here? Is it like a sudden jump in proplastid or amyloplast gene expression, or is it truly a synchronous process?
Do you have any way of mapping the progress of pseudo-time onto the progress of actual time?
So the question was about mapping the progress of pseudo-time along the progress of actual time. So yeah, that's something we want to do in the future. We want to actually verify this pseudo-time. We want to see if these cells truly are undifferentiated meristematic cells, and we want to see if this trajectory is actually correct, because without actually validating that, we can't truly make applications to the plant. So.
We will now also take questions from the audience. All right, thank you, Ahmad.
Thank you.